Welcome, everyone, to the Biophotonics Microscopy Summit. Thank you for joining us, and in this session, you will hear from Simon Bush, a product manager at Prior Scientific Instruments. Simon earned a master's degree researching plant cell biology and has over a decade of experience working in the microscopy field. Today, he's joining us to highlight modular microscopy for prototyping and discovery. If you have any questions or comments, you can type them right into the Summit chat box that is on your screen. And if you missed part of today's sessions, you can always come back later and watch them on demand. That's all for me. Simon, it's great to see you again, and we appreciate you taking the time to join us at the Microscopy Summit.
Thank you very much, Jacob. It's always a pleasure to contribute to these webinars. Hopefully we'll be able to present something new and different and exciting today, maybe a different side of Prior that not many people know. So today I'm here to talk about our modular microscopy platform, the OpenStand, and more specifically how it's helped some of our customers to realize their own ambitions for developing their own products. So that's why we call it precision at scale. Prior is known for its precision technology, but what we're really trying to do as one of our business areas these days is helping our customers to scale up their novel microscopy or other imaging modalities to produce the instruments of the future, really. We can help academics to spin out, or we can even help large companies also realize technology of their own. So our motivation really, as I said, with this approach to business is to help research only and novel microscopy modalities become more mainstream, to make automation the real standard for microscopy. Prior's bread and butter is microscopy automation. It makes sense for us to do this. But also there's also new and different types of motorization, more niche types of motorization for microscopy that isn't commercially available, and we try to make that type of technology accessible to people. We want to build design to production partnerships with our clients from prototype to final product, and you'll see how we do that in some of the content I've just coming up in the next slides. And use our expertise in production and other areas of microscopy business, the logistics, the compliance testing, and the supply chain setup that really fill gaps in our clients' knowledge to help them go from kind of an academic scenario into something that is more commercially facing. And then in the long term, we want to contribute to and share in the commercial success of our clients. I think a difference with Prior is that we're not only trying to sell you components, we're trying to build that long-term relationship. So you can rely on us to underpin the basics of your instrument, and you can market, the bits that make the technology especially exciting to the wider microscopy community. The main way that we start off projects like this is through our OpenStand platform. Now, I won't discuss the minutiae of all the different modules that you can get through this platform, but the concept here is that you can pick up motorized stages, motorized focus, laser autofocus, motorized fluorescence hardware, and even more obscure pieces of technology like motorized camera changers, light source inputs, and even things that maybe I or yourself in the audience haven't imagined just yet. Prior's design capabilities are extensive, and you can apply those motorized modules to upright microscopes, large format microscopes, inverted microscopes, and microscopes to address almost any application that you might be able to think of. And then we can take in also components from third-party suppliers that themselves are specialized in different types of technology, maybe camera technology or confocal microscopy technology, and combine the two together and then produce a very, very robust platform that can then be iterated on. Maybe you're designing an imaging technique that is completely new and bespoke and radical. Maybe you just need quite a simplistic instrument, but you're having a little bit trouble selecting some of the components, but you really need something customized and user-friendly so that your end user can interface well with your analysis package or your AI platform or your cloud-based image sharing solution. So we're just going to pop over into a video now that will kind of showcase behind the doors at Prior, both in the UK and globally, so you can see how we take our customers from ideas through to full production. So what you can see on screen here is an inverted open stand underpinned by all of the classical motorization technology that Prior is famous for. But it all starts here in our machine shop. More than half of our floor space at the UK site is machine shop, and this gives us control over all of the machined parts that we make. It allows us to design things from scratch really quickly. We've got machines that can run 24/7. We've got machines that can run in small batch sizes, and we've even got an R&D suite of tools as well that allow us to do this prototyping. Many of our engineers actually come from the machine shop, and that means that we've got this really nice interface between the beginnings of a product and the end of the product as well. We have a complementary suite of testing tools as well, so we can analyze everything we make, and then if something does need to be changed, we can rapidly deploy those changes rather than working with a third-party supplier. To follow on from there, we have an extensive R&D team with multiple different disciplines available to us, from mechanical, electronic, software, nano positioning as well, and optical design. Here you can see one of our test rigs down at our nano positioning site in Torquay in the UK. And this is our test room in the UK, where we come up with new and advanced imaging modalities for our systems. You can then take that design and translate it straight into production just at the other end of the building. This is our electronics suite, where we test all of our controllers. ProScan 3 has been a bread and butter solution for many major microscopy companies for a number of years now. And there's a huge diverse array of expertise within these rooms. We have everything from basic assembly all the way through to these very, very complex nano positioning solutions that you see on screen here, that require an extensive level of experience and testing, and a real in-depth knowledge of both the mechanics and the electronics and the software that underpins something so precise. We can really go in depth and troubleshoot and identify very, very specific requirements for different applications in a huge number of different fields. The net result is a huge array of products to choose from for different applications that can all be brought together in perhaps unexpected ways. And also we have the capacity to store these things on the shelf and roll them out in a way that allows our customers to supply their own customers in an efficient way. Prior's global reach is also a big factor in supporting our customers. We have 5 sites throughout the world, our head office in the UK, then a secondary manufacturing, design and sales office in the US, and then sales offices in Germany, China and Japan. And what this does is it means that no matter where you are in the world, we can support you with this sort of work. Now, for the rest of the presentation, I'm going to talk about 3 different clients of ours who very graciously allowed us to speak about them, and what technology they provide and their journey with us. So the first of these customers is Chip Nano Imaging. They're based in Norway, and they have developed a specific way of imaging samples at very, very high resolution, underpinned by TIRF. That's total internal reflection fluorescence microscopy. They produce photonic chip technology for holding and illuminating samples at high resolution with a massive field of view, which is what makes their technology unique. They're a spin out from the University of Tromsø, and the motivation behind their product was that they were getting significant demand for the use of an R&D instrument. So they've been able to take that research, that organic research, and translate it into a product. Underpinned by the research of Dr. Øystein Helle, and whilst initially funded by an EIC grant, they're now backed by corporate and technology transfer funds. So they're making that transition out of academia. Their solution, which Prior makes part of, is a combined super field of view TIRF, super-res, wide field fluorescence, and 3D deconvolution system. So all of these very exciting high-resolution fluorescence modalities. From us, they get motorized XYZ, motorized filter turret, nose piece, light source changer, which allows them to realize wide field fluorescence using a conventional multi-channel LED source, and also introduce a laser source to allow them to do different types of more advanced imaging. And then we can also introduce a hardware autofocus, which is really nicely suited to their chip technology, which I'll talk about in a second. They also use the very well-regarded Teledyne Photometrics kinetics camera on a T-mount to obviously give them that massive field of view that makes their technology so unique. The applications for this are not only research, but also commercial microbiology and non-clinical histological investigations. So their technology, which is called chip turf, works like this. Rather than having the excitation signal fluorescence pass through the objective that you also collect with, they have a complicated multi-axis mechanical setup that allows them to align their excitation objective with waveguides within a photonic chip. So they're using effectively semiconductor technology to do life science imaging. They can then collect the signal that goes down this waveguide and illuminates the sample directly via a separate objective, and that's what allows them to obtain this massive field of view. The way that TURF works is that you excite the sample not indirectly using an evanescent wave that is produced by fluorescence that's produced by light being totally internally reflected within some sort of guiding structure. In this case, it's the waveguide of the chip they produce. And that means that they are only illuminating a very, very thin section at the bottom of the samples. What the benefit of this is that if they avoid additional fluorescence from other planes that they're not interested in imaging, and that effectively gives them very, very good spatial resolution in the images that they acquire. I should also mention as well that the chips can be used for cell seeding as well, so you can prepare your samples directly on chip rather than having to take cells out of a culture flask and then seed them onto the chip prior to imaging. So you're at no risk of perturbing the sample during the preparation of the chip needed for imaging. These images give you an idea of the net result. On the left-hand side, you've got a comparison between chip turf and objective-based turf. You can see there from image C the massive size of the image that they're getting under this high magnification image. And then in box F, you can see a comparison of the flatness of field that they get from this image. Because light is approaching the sample from all different directions within the waveguide, they get very, very even illumination over this field of view in comparison to objective-based turf, where you get most of your intensity towards the center of the field of view, and then it drops off quite rapidly towards the edges. So you're getting a very wide field of view, but you're not compromising on the image quality at all. Then a natural way you can progress with this is you've got a very large field of view at high resolution. How can you get higher resolution? Well, you can apply computational super resolution techniques. So you can see here how they've taken just a conventional turf image and processed that using their proprietary Arctic storm technology to produce a high resolution image. And they've also used fluorescence fluctuation based super resolution microscopy as well, that looks at that statistically processes the temporal variations of the fluorescent signal that they collect. Finally, and perhaps even more excitingly, these chips are compatible with electron microscopy. Correlative microscopy was something that I did a small amount of during my research. I'm always excited to see this type of technology being applied. Because the cells can be directly seeded onto the chip and then imaged and then transferred into an SEM, and you've got a massive field of view to work with, the system that Chip Nano produce allows you to easily locate the features of interest that you want to look at and correlate those images together. And they're working to further establish that workflow as standard for the system that they make. So to talk a bit about the journey that Chip Nano had with us, They first contacted us in 2020, and we rapidly delivered them a prototype based on the open stand. They then sourced additional funding, and then we started to iterate on the design. We first went to an F-mount camera mount to allow them to achieve a bigger field of view. We optimized the system footprint to make the system smaller. We added the dual light source input to allow them to access a laser-based imaging modality as well as basic fluorescence in addition to their turf imaging on the chip. Then we further upgraded the field of view, and we're going through a process now with them of modularizing the system so that they can choose out exactly which modules they want from us for each customer and make each system unique to the laboratories that they go into. The next case study I'm going to talk about is how we made a more significant leap in development in a short space of time, how we went from open stand to something rather more unique for this customer, Hawk Biosystems, based in Spain. So Hawk Biosystems produce their own assay technology called QF Pro for quantifying proteomic events. It's underpinned by research from Dr. Veronique Calleja, who's now the CSO at Hawk, done at the CRIT. And it uses an adapted proprietary FRET film technology to collect data at an effective 10 nanometer resolution. This technology has been used in multiple clinical studies and been able to validate numerous CDX biomarkers. And they're now going through their commercialization cycle. And what they really needed was a way to apply this established QF Pro technology in an instrument that was easy to use, because FRET film is a very research-oriented imaging technique, and for an average microscopist, they might never have come across it. So they needed an instrument that would allow their technology to be increasingly accessible. So their system, the Violet 3, is a fully automated, dedicated FRET film system. It contains motorized XYZ control, a motorized filter turret, and light source changer. And we did a lot of work with them in creating this system, which is manufactured at Prior, to make sure that it was laser safe. We did help them with the compliance testing, and we also facilitated putting in interlocks for the laser system and the wider box, so that if anyone did open a panel that they weren't supposed to, they would be protected from the high power laser that's being used. The applications for this device are in research, drug development, and translation of research to the clinic. Allows you to qualify certain biomarkers as being clinically relevant. The benefits of their QF Pro technology are signal amplification that overcomes the autofluorescence inherent to many tissue samples, an extremely high signal-to-noise ratio and high specificity because of the labeling process that QF Pro allows. It allows you to locate spatially within a piece of tissue where these interactions are occurring. It's not just an implied interface between two proteins that you take from a destroyed sample. You can actually see it in the tissue, and you can get quantitative results, not just that spatial information. You can actually quantify the level of interaction, which I'll talk about in a minute. You can measure protein-protein interactions. You can measure drug and receptor interactions. You can investigate post-translational modifications with this technology, and also look at protein DNA interactions. So just to discuss a couple of the terms that I've been using. So FRET is fluorescence resonance energy transfer, and that is the transfer of light from a donor molecule to a receptor molecule, and it is that transfer of light that can only occur over a 10 nanometer range, and thus illustrates that there is an interaction going on. Any further apart, you wouldn't get any of that donation of light. So what they're actually looking for is the emission of fluorescence light from an acceptor molecule. And then FLIM is fluorescence lifetime imaging microscopy. This looks at the fluorescence decay of the donor and acceptor molecules and allows you to quantify the level of interaction based on how the lifetime changes for your donor molecule. This is discussed on this slide in a little bit more detail. So the FRET efficiency is the fraction of energy transferred per electron excited in the donor, how much of that is transferred to the acceptor molecule. As I said before, it only occurs when the acceptor and donor are within 10 nanometers of each other. And thus that's what proves out and what allows Hawk to claim that they're achieving a spatial resolution of 10 nanometers for these interactions. In order to make the technology even more accessible, they have a patented amplification process. The HRP molecules there enable the activation of fluorescently labeled tyramide molecules, which then bind to the primary, secondary antibodies that they use when staining the sample, and also adjacent tyrosine residues within the tissue. This amplification means that the system becomes a lot more user-friendly, and also means that rare and difficult to identify biomarkers can be observed spatially within the image. And these two images give you a representation of how these images actually come out. You don't get the multicolored image that you see here is not different fluorescence channels, rather it is the different lifetimes of the molecules that they're investigating, so they can see based on the intensity of the signal where the interaction that they're trying to observe is strong or weak within the tissue section that they're observing. So to quickly look at a case study, they investigated the interaction between PD-1 and PD-L1 in non-small cell lung cancer. This is a typical assay that's used for investigating whether a patient is likely to respond to drug treatment, and it's all underpinned by the interaction between PD-L1, which is expressed by cancer cells, and PD-1, which is expressed by T cells who are looking to destroy these cancer cells within the tissue. The interaction of these two proteins is a good indicator of drug response, but what the next slide I'm going to show you will show is that the presence of just one of these markers alone is not a good indicator. So let's take a quick look at that. You can see here that they're looking at just the detection score for PD-L1, that's the protein expressed by the tumor cells, versus the score that QF Pro gives you. And the result of this data is that by a metric of probability of survival a number of months after initial treatment, by looking at the interaction between the two markers, you get a better indication of survivability than just looking at one of the markers alone. This means that patients can be directed towards an alternative treatment if they're unlikely to benefit from a typical drug treatment that would only look at PD-L1 score, which might mean the difference between survival or further complications. So Hawk's timeline, Hawk's journey with us, they started again with an open stand solution with a very minor modification to allow them to introduce the laser source that they use for their FRET FLIM imaging, and also a wide field fluorescent source. Because of the good results that they had with that system, they were able to get further funding, and that allowed them to place an order for 2 prototype systems. One of those systems we delivered extremely rapidly to allow them to further explore the technology in-house with the new system, and the other one was taken away for compliance testing, and that allowed us to ensure that by the time they were ready to transfer into production later last year, the system was laser safe, it was electronically compliant, and it was also compliant for the markets they were interested in accessing. They've shifted into production and are now delivering units to their key markets. Our final case study today is the is for Sparta Biodiscovery and their AGIS-1 system. This is probably the most complex of the three as well, so hopefully some of you are hanging on for something really advanced. Sparta Biodiscovery are a company that spun out of the Stevens Group at Imperial College London, and their goal was to develop tools for nanoformulation analysis and quality control for next generation pharmaceuticals, again in an easy to use, accessible instrument that could be transferred into a clinical setting. And really, as a testament to the success that they've had, they now have a partnership with Sartorius, a major company in that space. You might be wondering why they're called Sparta. Well, it's an acronym for the technology that they developed. The Sparta stands for Single Particle Automated Raman Trapping Analysis. It uses a 785 nanometer laser for trapping of individual nanoparticles, and they can do this in an automated fashion. They've got a fully bespoke and customized detector assembly of their own design, and what they needed us to do, again, was to integrate that with XYZ motorization and a casework that would be compliant for their international delivery goals. The applications for this technology are, again, research, drug discovery, and gene therapy. The technology is extremely capable. It can work with a number of different samples in sample types in all different types of media. But how does laser trapping work? So laser trapping relies on a highly focused laser beam to hold and move samples around. The laser light can apply sufficient force to resist random movements of samples in media or air or gravity and isolate these particles and keep them static while they're being analyzed. Sparta can automatically detect and grab nanoparticles and other microscopic samples for analysis. And being able to do this means that the AGIS-1 can analyze a very large number of samples and get a statistically significant amount of data in a shorter period of time than is currently possible. And the analysis that they can do is not just on the composition. They can also infer the functionality of different types of nanoparticles. They can do size measurements, and they can also do temporal measurements as well. They can look at how a nanoparticle might behave in different media over time, because they can hold the particle static under the optics and collect data that way. So the data that the system processes and puts out is extremely rich and also provides a huge number of data points. So you can start to make conclusions that maybe previously weren't possible to make. That means that you can more quickly dismiss avenues of investigation for drug development that aren't of any use, and also produce much more robust data when trying to support a translation into a clinical trial. So there's a couple of case studies that I'll just talk about here. So the first of these is looking at the formulation of the drug doxorubicin. It's worth mentioning you can have a read of all of this on Sparta's website if you'd like more information. So what they were looking at here is the size and loading of different nanoparticles when making up this drug formula. And what was important here was looking at the diversity of the types of molecules that you're creating, and whether having a larger particle had any advantage in terms of drug delivery. Could you have a lower hypothetical dose of a drug but still deliver the right amount in a targeted place? What they found through their automated imaging and analysis was that increasing the size of the nanoparticle didn't significantly increase the drug loading. And what that effectively means is that the producer may be able to refine the process of creating the drug characters to ensure the optimal size, so they don't waste materials or end up with batches that are maybe less effective than they should be. Second case study, again, looking at cancer biomarkers. So again, because they're able to look at the size and the chemical makeup of all the nanoparticles that they look at, they're able to come up with very specific profiles of the different types of components within these. So in this case study, they looked at exosomes or extracellular vesicles, which are an actively studied class of cell-derived nanoparticles, which have a lot of promise for acting as biomarkers for different types of cancer. Their system allowed them to generate detailed fingerprints of individual exosomes, without which they wouldn't be able to produce the three-dimensional graph that you see on the right-hand side there. What they've been able to do is, by quantifying the specific profiles of different materials that they have within the exosomes, is to create a very clear binary data set here, where they're able to differentiate between non-cancerous and cancerous cells purely based on the extracellular vesicles that are being excreted from the cells. Their timeline with us was more of a long journey. So their technology was really unique, and that requires additional funding, and it requires more convincing of different partners that your technology is a good avenue to go down. So they started with us in 2020. We integrated their custom laser system with our mechanics and had a prototype ready for them a year later. We did an on-site demo, and then we went through a long period of them going away and waiting for funding. Now, Prior really likes to help its clients. It really likes to support them. So we kept talking. We kept talking about different ways that we might develop the system, any improvements we might want to make in production. And then, because of the good work of the team at Sparta Biodiscovery, they were able to source that additional funding and ordered additional beta units from us. And then we're able to secure that relationship with Sartorius and have since moved into significant production with Prior. So it's a really good example of someone coming with a unique technology that needed to be underpinned by robust, proven mechanics from a known supplier in Prior. And then they've come through that journey with us over a number of years, supported all the way, and now they've made it through to that full production. They've been able to utilize the manufacturing facilities that I showed you at the beginning of the talk. If any of this is of interest to you, if you feel like you might have a technology that you want to spin out, or maybe you know somebody at the lab you work out that does, please direct them to our website or ask them to contact us. We've got a huge amount of additional information on all of this on our website, on our new website now. And you can also explore how you can work through other ways of prototyping. Maybe you've got an instrument in your lab that needs upgrading and you're just in the very early stages of your technology development. Prior can also support with that sort of thing. So that's my presentation. I hope you enjoyed it, and I'd be happy to receive any questions.
Thank you so much, Simon, for joining us to help viewers achieve precision at scale with modular microscopy. Just to echo your statement to everyone that's joining us in the audience, if you haven't already, you can type your questions and comments into the Summit chat box, and do not worry if you've not been answered yet, you will be reached via email at a later date. This session is hosted by us at Photonics Media, and we hope you're enjoying the inaugural Biophotonics Summit. Thank you all so much for joining us.